Let's continue with the organic molecules. By definition, organic molecules are those that contain carbon and hydrogen. Carbon and hydrogen. And now carbon is number 6 on the periodic chart of the elements. Therefore, it has 6 neutrons, 6 protons, mass assignment is 12, and that means we have also 6 electrons. So that means we're going to put 2 electrons on the inner shell and 4 on the outer shell. The outer shell, the second shell, can hold 4 more electrons. Well, how likely is it to get somewhere 4 electrons? It's not very likely. Giving up 4 is not very likely either. So we're right in the middle here. And that leads to the unique properties of organic molecules in which carbon tends to bond with itself and form these long chains and ring structures. You have some examples right here. So hexane or cyclohexane forming circles. You have to consider every corner of this hexagon is a carbon. That's not printed there anymore. But so we'll take a look at some typical organic molecules. So they're characterized by these chains or ring structures where carbon bonds with itself, and you see generally 4 lines surrounding each carbon, which means each line is a pair of shared electrons for a covalent bond. The unique properties of the organic molecules are due to these functional groups, and here's one, the carbonyl group. That one is pretty important. Then here we have the alcohol group, OH, and unfortunately it's a little mistake. This should be only 1 line right there, not 2. The sulfhydryl group, we'll not be talking too much about that one. Again, there's a little mistake here in that figure. And the amino group is very important, also only just 1 line right here. And then we have the carboxyl group. That one is pretty important. So these weak organic acids, and then the phosphate group right there also. There, this is actually okay. So what you should know, the carbonyl group, the hydroxyl alcohol group, sulfhydryl group, definitely the amino group, carboxyl group. So these are some of the functional groups that you should review and take a look at. Some other interesting things about organic chemistry is that a lot of times we see stereoisomers. That means these are molecules that are mirror images of one another. And it's kind of interesting that only one of the mirror images is usually biologically active. So take a look at your right hand and your left hand. They're not identical, but they are mirror images of one another. And so we call the stereoisomers, they're either right-handed or left-handed. So D for dexter from the Latin for right, or L, I guess you can do left, that works. So these are the 2 versions, or the stereoisomers of a compound. And biologically active is just only one or the other. And so biologically active would be, for example, the L amino acids and the D sugars. Okay, so we'll start with a large group of organic molecules known as carbohydrates. We eat a lot of them usually, so they are sugars and starches. So we're going to take a look at how they are, how we get these carbohydrates. So they are composed of carbon for carbo, and then hydrates with water. Now it's not exactly water, but if you take a look at, let's say on B right here, you can see that each carbon is surrounded by 2 hydrogens and an oxygen. So technically an H2O, 2 hydrogens, 1 oxygen. So the summary formula is CnH2nOn. So the same amount of carbon as oxygen and twice the amount of hydrogen. Those are carbohydrates. We'll start with the simple ones. Those are called monosaccharides, or simple sugars. Some examples are given here: glucose, fructose, galactose. You have them over here as well. They come either as a linear molecule or as a ring structure. This is actually equivalent. These are the same. And so these are monosaccharides, or simple sugars. Simple sugars. And we'll test some of them in the lab. Then when you're connecting 2 simple sugars together, you'll get a disaccharide. 2 simple sugars together, dis, 2 monosaccharides joined covalently. And some examples here would be household sugar, sucrose, and then also lactose, milk sugar, and maltose. Now, if we're connecting many of these simple sugars, then we'll get polysaccharides, and those are your typical starches. So here we have all sorts of starches, whether corn starch, potato starch, you name it. And these carbohydrates, they are great for storing energy. This is what your cells really prefer to burn for energy to create ATP. Okay, now let's take a look at a general theme that we see in organic chemistry. How do we go from a simple building block to multiples? And so the formation of disaccharides here as a example. How do we go from connecting two single sugars into a single disaccharide? And there is a principle that you will see throughout organic chemistry, not just for the carbohydrates, but also for lipids, for nucleic acids, and for proteins. That's called dehydration synthesis. And dehydration means take water out. So we're putting building blocks together by taking water out. And let me show you how this works. Of course, this requires specific enzymes, but here's the formation of a disaccharide in the next image right here. So we'll have two glucose molecules, 1, 2 here, right next to one another, and we're going to bring them in close proximity. We're going to take the H2O out. The H2O goes right here at the end. We're going to connect the two sugar molecules here with this glycosidic oxygen bridge, and now we have maltose. So we have two sugar molecules together. And here we are forming household sugar. We're going to take glucose and fructose. We're going to take H2O out. So basically right here. And then this water is right here, the H2O. And then we're going to have the sucrose. So we're going to have the sucrose molecule right here. Okay. And now if we wanted to break down these larger polysaccharides, for example, and make them simple sugars again, guess what? We're going to put the water right back in, and then we'll break these larger complex building molecules that are composed of many building blocks. We're going to break them down into the simple ones again, and that is known as hydrolysis. Definitely a term that you need to know. Hydrolysis, adding water to break things apart. So if we have a starch molecule here with many connected simple sugars, we add the water, and then that means now we're breaking it down into two maltose molecules, for example. So hydrolysis and dehydration synthesis are exactly opposite, and you should know both. So you need to know that. Hydrolysis is also often referred to as digestion reaction, because if you're eating, let's say, a piece of bread, you want to break it down into simple sugars. Well, how do you do that? Hydrolysis is the answer. Okay, we're moving on. We're ready to go into lipids. Lipids is a very diverse group of organic molecules. The only thing that they have in common is they're all hydrophobic. They don't like water. They don't like to mix with water. They have no polarity, and so all of them are hydrophobic. So they are insoluble in any kind of polar solvents such as water. They mostly have hydrocarbon chains, so mostly carbon and hydrogen. And we'll start with our dietary fats. Those are triglycerides. Triglycerides, tri refers to 3. So what do we have 3 of? Well, we have 3 fatty acid tails that are connected to one glycerol molecule. So we have glycerol and 3 fatty acids being connected by dehydration synthesis, actually. And then there are different versions of these triglycerides. We have saturated fats. That saturated means that we have the maximum amount of hydrogen added to it. Unsaturated means we have some double bonds in the molecule that allow then for not the maximum amount of hydrogen. So let's take a look at this. Here's some fatty acids as an example. Here we have palmitic acid. It's a saturated fatty acid. You see that there's the maximum amount of hydrogens along this hydrocarbon chain right here. No double bonds, and every carbon has these 2 hydrogens. Now here we have, all of a sudden, we have a double bond. And that means now these this carbon here no longer has room for extra hydrogens right here. So we're not saturated with hydrogen anymore. So we are unsaturated. So in this case, we have a mono unsaturated fatty acid. Here we have another example where we have a double bond between carbon here, here, and here. And that you see all the missing hydrogens right there, because remember, every carbon can only have 4 covalent bonds. If you take up 2 right here and a third one to another carbon right here, then you now have room for 1 hydrogen. So this here is a polyunsaturated fatty acid. So here's how the triglycerides are formed. So that means we have a glycerol backbone right here. This is your glycerol backbone. We're going to connect 3 fatty acid tails by dehydration synthesis. We're going to take 3 waters out. We're going to connect 3 fatty acid tails. Here are our 3 waters, and that is a triglyceride. A little sidetrack here about keto bodies. You may have heard about the keto diet. It's pretty popular these days. That means that we are breaking down fatty acid tails to generate energy. So we're doing the hydrolysis of triglycerides, and then we're releasing these fatty acids, and these fatty acids then will be broken down into keto kind of containing compounds, smaller 2-3 carbon pieces that then enter the Krebs cycle for energy production. So basically a keto diet is a diet that forces your body to, or your cells, to burn fatty acids for energy rather than relying on carbohydrates, which your cells tend to prefer. But if you don't eating much or of any carbohydrates, then you're going to force your body to produce keto bodies. Generally, that's not a big problem, but if you are, let's say you're diabetic and you will lack insulin and your cells cannot pick up any glycerol or any kind of carbohydrates that is in circulation, then your cells have no choice but to rely exclusively on fatty acid metabolism. And that can lead to a problem because too many keto bodies can lead to something like ketoacidosis. And that is what you see in uncontrolled diabetes. If somebody is in ketoacidosis, they're near a coma. So it can be pretty dangerous. But it will be an increase in keto bodies in the blood, and they can lower the pH, and then you have this ketoacidosis. So these people will be hyperventilating to try to breathe out some CO2, which we just talked about in the other lecture on general chemistry, that the CO2 carbonic acid buffer system. But anyway, if you just go on a keto diet, chances are you're not going to go into ketoacidosis because you're going to still have some carbohydrates, and you definitely have some insulin to put those carbs into your cells. Anyway, there are more lipids that we should talk about, namely the phospholipids. They form the main backbone for your phospholipid bilayer, and so they're very important. They are amphipathic. That means they're dual natured. They have a non-polar region of the molecule, and they have a polar region. So the non-polar end is hydrophobic, and the polar end of the molecule is hydrophilic. It makes them dual natured, and it's perfect if you want to form a barrier. And we'll talk much more about biological membranes. Lecithin is not that important for us. But right here, phospholipids. Here we are. So we have the polar head group, that's this part right here, and then these 2 squiggly tails right here, that will be the non-polar hydrophobic portion of the molecule. If you want to add more detail, you can put this complex organic polar head group right here, and then here would be your 2 fatty acid tails. Most of the time people will just use a little diagram, and it will show something like this. You have a little head, and then 2 fatty acid tails, and another head, 2 fatty acid tails. And if you connect many of them, you end up creating a phospholipid bilayer, and we're going to talk much more about that. Now let's take a look at steroids. That's another lipid category, and they all have the same backbone structure. It's 3 6-carbon rings and then 1 5-carbon ring, and then various attachments. Cholesterol is your model molecule right here for a steroid. And let's take a look at this. Steroids, that's what they look like. So they have this they're shaded in green and yellowish. You have the steroid backbone. So you have 3 6 carbon rings. So this is 1, 2, 3, and then 1 5 carbon ring, and then various attachments. So this happens to be cholesterol. This is testosterone, the male sex hormone, and then we have estrogen, the female sex hormone, and here's cortisol, your stress hormone. I wonder what happened to my pen. Okay, so moving on to prostaglandins, actually. That's a little sidetrack here. It's also, these are lipid molecules. They're acting as local messengers. They're often overlooked, but they play an important role as local messengers. So we call them also tissue hormones. You have various forms of prostaglandins right here. Prostaglandin E1, E2, and so forth. What you can see here is a fatty acid tail that starts to kink over and then sort of flip over and return on the other side, make a little U-turn right there. Again, these are tissue hormones. They have various regulatory functions as well. A lot of times they're pain mediators, which we don't like very much, but they lead to massive vasodilation and sort of mediate inflammation and inflammatory processes. Moving on to proteins right now. So proteins are composed of amino acids. Now, what's an amino acid? Okay, so an amino acid is a relatively small organic molecule that has an amino end on one end and a carboxylic acid or organic acid part on the other end. And in between, there's another carbon, and that one can have various side chains that we call R groups right here. So this would be your R groups right there. And I think it's probably easier to take a look at one of these images right here. So here's a typical amino acid where you have the amino end right here, the carboxylic acid group on the other end. In the middle, you have a carbon that contains a hydrogen and an R group. And this R group can change. R means just variable. It's a residual. We can have here different types of attachments for this R group. Here are some examples given. This also tells you about the naming. So each amino acid, and there's 20 that form all of the different proteins in the human body. So here's arginine, the 3-letter abbreviation ARG, and then the 1-letter symbol R. Or let's take another example, cysteine right here, 3-letter abbreviation CIS, and then the 1-letter abbreviation, the international code is C. So when you see a paper or somebody publishes a protein sequence, they will only use the 1-letter symbol. So these 1-letter symbols are pretty useful because it denotes an amino acid and everyone knows which one that is. Okay, so again, the amino acids, there are these 20 amino acids. They all have an amino group, that's the NH2 right here. They have a carboxylic acid group, that's right here, weak organic acid. And in between, they have a functional group, that's with this R group. And this R can vary. It can be like a valine, look like this here, a tyrosine, fairly large kind of group, cysteine with a sulfur group right there, arginine. So there's different ways, and lucky for you, I'm not going to make you memorize them. But anyway, so let's take a look at proteins. Proteins are polymers of these amino acids. How do you connect the amino acids? Well, again, by dehydration synthesis. In this case, we call the bond between each amino acid, we call it a peptide bond. That's kind of important. And as soon as you're connecting a few of these amino acids, you have a little peptide. Once the amino acid chain gets longer, and then we have a polypeptide, and eventually we're going to call it a protein. So depending on the length, you have the various kind of names. Now, these amino acids, these complex amino acid type of organic molecules, they end up forming hydrogen bonds that then give them secondary and tertiary and even quaternary structures. So let's take a look at the sort of organizational levels. The primary structure is just the amino acid sequence. The secondary structure is formed by these hydrogen bonds, and it's typically alpha helices and beta sheets. So let's take a look at this here. So we have the primary structure, that is just a polypeptide chain. So just amino acids connected to one another. And then due to hydrogen bonding, we get alpha helices, we get beta pleated sheets, and those are the secondary structures. Both of them are secondary structures. And once we put these secondary structures together, then we get a tertiary structure. So here would be an example, the heme group. And if we have several subunits in a molecule, in a protein, such as the hemoglobin, we have 2 alpha chains and 2 beta chains. Now we have a quaternary structure because that denotes the entire 3-dimensional arrangement of all the different polypeptide chains with respect to each other. So the tertiary structure is putting all the alpha helices and beta pleated sheets and all the linker regions, whatever you have there, put it together in 3-dimensional space. So we have a 3-dimensional shape structure. The quaternary structure means that we have various subunits that come together to form a fully functioning protein. So here, nice picture of the hemoglobin. There will be 2 alpha and 2 beta chains. And our final group of organic molecules is the nucleic acids. They form, of course, our famous DNA and RNA molecules. All databases for DNA and RNA. Nucleotides are the building block for DNA and RNA. And so here we have one of these building blocks. This would be a nucleotide. It always has a phosphate group, 5-carbon sugar, either ribose or deoxyribose, and a base. The base can have 4 different flavors, I would say. We can have guanine, thymine, cytosine, and adenine. For RNA, we're going to replace the thymine with uracil. But for now, let's take a look at DNA first. So we have these nucleotides. They are always composed of a 5-carbon sugar, phosphate group, and a nitrogenous base. Any of these 4 different nitrogenous bases. The nitrogenous bases, there are 2 out of 4 that are a little bit larger, and the other 2 are a little bit smaller. So the pyrimidine ones, these are single ring carbon structures, so that would be the smaller ones. That's the thymine and the cytosine. You can see how they're a little bit smaller right here, the C and the T. And then the purines, those are 2-ring carbon structures. They're a little bit bigger, and you can see the G and the A, they're a little bit bigger circles right there. Here, nice and big. A nucleotide always composed of phosphate, 5-carbon sugar, ribose or deoxyribose, and a nitrogenous base. Okay, either with 2 rings or with just 1 ring structure. And so here the nitrogenous bases one more time. So that would be here, we have the purines, the bigger ones, that's adenine and guanine, and then the pyrimidines, that would be cytosine and thymine for DNA. And if you want to look at RNA, for RNA, you need a uracil. So let's take a look at how this is connected. We're going to connect these building blocks, these nucleotides, by dehydration synthesis as before. And so we can form very long strands of nucleotides. Now in DNA, we have 2 strands of nucleotides that face one another, and they're connected by base pairing rules. So that's the basis of the genetic code right here. And of course, you already know that the deoxyribose is covalently bonded to one of 4 bases. So we have the adenine, guanine, cytosine, and thymine, the purines and the pyrimidines that are listed right here. And then each one of these bases can form hydrogen bonds with other bases, but there are base pairing rules. Guanine forms hydrogen bonds with cytosine. How many? 1, 2, 3. 3 hydrogen bonds right here. Adenine and thymine can also hydrogen bond with one another, but only 1 to 2 hydrogen bonds. That means the bond between guanine and cytosine is a little bit stronger because we have 3 hydrogen bonds. Here we have only 2. Okay. Let's take a look at the difference here with RNA. RNA has, it forms, it uses the sugar, the 5-carbon sugar ribose. So by comparison, DNA nucleotides, they contain deoxyribose, and RNA nucleotides, they contain ribose. Minor, very small change, this H versus this OH right here. In RNA, we will find the base uracil, and it will replace the base thymine, okay? So another difference is that RNA is typically single-stranded, a single chain of nucleotides, and DNA is usually double-stranded, 2 strands that are connected by base pairing rules, by these hydrogen bondings. And ribose will covalently bond to 4 different bases, same bases except for the thymine. We don't find any thymine in RNA. We have the uracil. So we have adenine, guanine, cytosine, and uracil. Okay, this is the end of our organic introduction to organic molecules. These are the molecules that you will need for this course. So the carbohydrates, simple sugars, and then the starches, the polysaccharides. Then we had the lipids, very diverse group. Then we have the proteins connected. We have these 20 amino acids that are connecting to form these polypeptides and proteins. And finally, we had the nucleic acids, DNA and RNA, just a basic overview. But take a look at that. I will post some worksheets to help you sort of work through these problems and use them as needed. They're optional, of course, and you should just use them as needed. Some of you have better background in chemistry than others, so use them as needed.